Hello everybody. Today I'm going to teach you about proposal writing. This is the most important lecture of all the lectures I have posted for you on YouTube up to this point, I should say. So the first thing is you should know a little bit about some formatting rules for your proposal. Everything should be written in English, should be in an 8.5 by 11 inch sided sheets of paper. Yours is going to be electronically submitted, but if you had to print it, it should be an 8.5 by 11 single-sided sheets of paper, no dual-sided printing, with page numbers at the bottom. You should have a page margin of 1 inch on each side, and you should use 12 point or larger. But I would go with 12 point, Arial or Times New Roman font for the body of the report. Remember, captions accompanying pictures and graphs, as well as citations for references, may be single-spaced and in smaller point size. And a lot of times, those are in italics too. And if you want to get a sense of what is standard, you should look at your background research articles to see what they do. So this list is extremely important, the components of a grant proposal. The first is the title, including your names, you're the authors. The second section will be the abstract, which you will write last. Abstract is a summary of the entire proposal. You write, or a paper, you write this last. Next will be the introduction, which will contain the objective and hypothesis. The next will be the materials and methods section. Material methods and proposed analysis. Proposed analysis could be inside the materials and methods, but in the YSAP grant they want a separate section, so we have to gauge it or judge it on a project by project basis. But the proposed analysis includes statistical analysis. Then you're going to have a reference section. Then you're going to have a separate list of the materials you're going to use and how much they cost. And you can even make a spreadsheet in Excel to show that. And then appendixes if needed. The title should be concise, descriptive, and informative. You have to include the independent variable and the dependent variable. And then if you're using any type of organism or reagents, well, if you're going to use an organism, make sure you mention the genus and species. Remember, the genus and species of any organism should be italicized. And any chemical, be specific about the chemical name. Next, the introduction should discuss the entity and why you're studying it, talk about the independent variable, talk about the dependent variable, and discuss the relationship between the independent variable and dependent variable and why this relationship is important. You really need to state in your project, why is your project important? Why does the YSAP want to fund your project? Why is this relationship important? Why is your project important? How does your project affect society? Why is your project important in helping society? And then at the end, as all research papers have, your objective and your hypothesis. Stating your objective. The objective of the study is to determine, let's say, echinacea on the regeneration of planaria or the regeneration rate of planaria. So that's the kind of thing. Your objective is going to include your independent variable and your dependent variable. And then stating a hypothesis, refer to the hypothesis lecture to really learn how to write a hypothesis. But just to quickly, you could say something like this: a change in the IV will cause a change in the DV. So if echinacea's concentrations increased, the regeneration rate of planaria will go up. Or you have to create two hypotheses. A null hypothesis: if planaria is given echinacea, then the regeneration rate will be no different than the control. So there will be no difference between the groups. And an alternative hypothesis, H1, which would say if planaria receive echinacea, then their regeneration rate or the regeneration or growth rate will be higher than the controls. That would be a situation where there would be a difference. And then you test this with a t-test or a z-test, depending on the sample size. Also remember, you can create hypotheses between two treatment groups also, and those treatment groups and the controls. This slide is in our Lincoln class page. It's a design of a grant proposal and a science research paper. As you can see, The relationship between the independent and dependent variable is an overlap, like in a Venn diagram. That overlap is what allows you to generate an objective and a hypothesis. Remember, both in a grant proposal and a science research paper, you have a materials and methods section or the experimental design section. When you write a proposal, you have data collection and analysis. How are you going to collect your data? The tables and graphs you plan to use, the statistics you plan to use, how will you interpret the data once it is collected? Whereas when you write a paper after all the data is collected, you have a results section which represents the data, and then a discussion section. Is the objective and hypothesis in the discussion of the paper the same as in the introduction? Does the data support or reject the hypothesis? Why or why not? How do the findings in this paper compare to those in the literature? Are there any experimental design errors explained? You should also talk about what are the limitations of your study. What are they? Why are they important? Why is the research in the paper significant? What is the overall bigger picture? And what future studies are you considering doing, and why are you considering them? And also you want to know what's the overall significance of your project too. How does it impact society? Materials and methods. The purpose is to describe how the experimental protocols will be carried out. This should be written in detail so that it can be replicated by another person. I want you to pay careful attention later on in the lecture to the 12-step method for writing materials and methods. That is what you should really use. Before writing the materials and methods, refer to your background research. It will tell you how to design your experimental protocols. Run some pre-trials. I know most of you are unable to do this before you're funded, so do it after. It will help you streamline and modify your protocols and help you learn how to generate your workflow. Use the narrative approach in the materials and methods section. You want to do it in writing. Do not write it in a step-by-step fashion. I don't like it that way. Write a paragraph discussing how the experiment will be set up. What preparation will need to be done before the experiment can be started. Describe any pre-trials that were used to help determine the methods. If you did a pre-trial, I've had students do it too. Do pre-trials, you're able to talk about what you did and how you modified your experiments. When you do your final paper, you don't necessarily have to talk about pre-trials when you write your final paper. I think you should, once you solidified or modified your protocol, then that will be the last, and that protocol is the one that you collected all your data. That will be the last one that you will include in your paper, and you don't have to talk about the pre-trials. And then how will the entities be labeled or organized, and what items and tools will be used to structure the setup. Continuing, if the data collection method will be repeated, tell the reader how often and with what experimental groups the methods will be used. For example, the procedure will be repeated for a total of 4 trials for each amount: 0 milliliters, 50 milliliters, 30 milliliters, and 45 milliliters. Another question you should be asking yourself is, how often should I collect data? How many trials do I have to do with each group? The frequency of data collection and the number of trials. So the first thing is the variation in the entity being studied. Remember, the more data or trials you have, the more accurate the results. So it's kind of like you have to make a judgment call. I will help you make that judgment call. So the variation in the entity being studied, results in making you think you have to do as many trials as possible, and then the speed at which the data are available, right? Your background research and your pre-trials should help you make this determination. Speed of the data collection is another determining or limiting factor. For example, if you grow plants with a life cycle of 30 days, you can't collect data, and your data collection only occurs monthly. So you wouldn't be able to collect that much data than if you grew bacteria, which grows every 24 to 48 hours. You'll be able to do as many runs as you want because in every 2 days you're getting data. Describing your data collection. So you got to answer these questions to describe your data collection. How will the data be collected? How will measurements be taken? How will the data be measured or obtained? What tools and techniques will be used? How will qualitative observations be recorded, if applicable? Remember, I don't really care much about qualitative data. You really want quantitative. 6, how often will the data be collected and recorded? How long will the experiment last? How does your research design address potential extraneous variables? And you should address each extraneous variable. Describing your data collection continued. Describe the hourly, daily, weekly, or monthly tasks that you will need to complete to maintain the environment in which the experiment is taking place. What measurements or observations, such as temperature, humidity, will you record throughout the experiment? These things could be the constants. Remember, when you're growing plants, you're growing fish, you're growing organisms, all the environmental conditions need to be constant. You need to discuss how you're going to maintain. Like, for example, you're writing materials and methods and you're culturing zebrafish. You need to have a separate section on the culturing of zebrafish. Talk about what temperature the water needs to be, how much food, what type of food, how many times a day is it going to be administered, where the fish came from, how they will be maintained, what we will look for, their location, how much sunlight. That's a separate section. That's a section in your materials and methods with a subheading, culturing zebrafish. And you would have to entertain all of these types of things. You should understand that you're going to rewrite the materials and methods section several times, and that's why you need to shoot it to me or send it to me as soon as possible. 12. Now this comes from an editing group in San Francisco. 12-step method of writing materials and methods section. This is a great list of things you need to have. So the first thing is order your procedures chronologically, meaning you want to order the different experiment protocols, or you want to write your procedures in chronological order or by type of procedure, and then chronologically within type of procedure use subheadings where appropriate to clarify what you did. I want you guys all using subheadings. Culturing of the zebrafish, experimental design, which we'll talk about. Let's go back to the planaria example. Culturing of planaria, a whole paragraph on that to subheading. Next subheading, cutting the planaria up, right? How you're going to cut it, with what kind of scalpel, what kind of knife, etc., etc., etc. The preparation of the echinacea, the concentrations of echinacea. Then you could do it like experimental design subheading. How you gonna the control group versus the experimental group. How much the concentrations of echinacea that are going to be subjected to the planaria. Then you would talk about how you're going to measure the growth rate or the regeneration rate of the planaria. And then you would have a data analysis section subheaded, and you would talk about you use t-test, z-test, why not. Also, that's what I forgot to say. You also want to discuss how many trials, how many runs for each concentration and for each condition. Okay? All right, so number 2, use the past tense and the third person to describe what you did. For example, the sample was incubated at 37 degrees for 3 days. Don't not say I incubate the sample at 37 degrees for 3 days. It has to be in the past tense. And the third person. I want everything in the third person. Okay? Describe your experiment design clearly, including the hypothesis you tested, variable measures, and how many replicates, meaning how many times you're going to do it. And you had your controls, your treatments, etc. Explain why each procedure was done. Reference may be made to a published paper as an alternative to describing a lengthy procedure. I want you guys writing it because you guys need to gain experience in writing procedures. Identify the source of any specific type of equipment, a specific enzyme, organism, or culture from a particular supplier which is critical to the success of the experiment. Describe in detail any modification to equipment or equipment constructed specifically for the study, and if pertinent, provide illustrations of the modifications. Precisely quantify measurements, all metric, and include errors of measurement. Right? So that was errors of measurement could be standard deviation also. The standard deviation, you do the mean and you do the standard deviation. 8. Describe the dates and sites where your field study was conducted, including physical and biological characteristics of the site if pertinent to the study's objectives. 9. Identify treatments using the variables or treatment name rather than an ambiguous generic name or number. You would use healthy donors instead of group 1. That would be your experimental group. Healthy donors actually would be probably your control. If required by the journal, mention the approval for the study by the relevant ethics committees and informed consent of the subject. So animal control, ethics, IRB, which is internal review board, etc. Describe statistical tests and the comparisons made. Ordinary statistical methods should be used without comment. In your case, you're going to use them because you don't have enough a lot of experience. So you should be even, like if you say you're going to take the mean and standard deviation, you need to state that you're going to do that. Okay, advanced or unusual methods may require a literature citation. Okay, show your materials and methods to a colleague and ask whether they would have difficulty in repeating your study. In a grant proposal, you need to be writing this materials and methods section in the present. 25 milliliters, I mean, in the future, I should say. 25 milliliters will be put in the test tube. When you finish your project and you write your paper, it all needs to be converted to the past tense. Please take heed of that. You're going to be writing this in the future. When you finish your project, you're going to convert what you wrote with all the modifications to the past tense. 25 milliliters was placed in the test tube, was incubated at 37 degrees Celsius, and then let set for 24 to 72 hours, something like that. Other points to consider when writing the materials and methods. You do not want to mix the results with procedures. That happens sometimes. Do not do that. Omit all explanatory information and background. Save it for the discussion. Do not include information that is irrelevant to the reader, such as what color ice bucket you used or which individual logged in the data. So this came from Science, Medical, and General Proofreading and Editing. It's a company in San Francisco. This is a great source. Really hones in what you need to know for the materials and methods. The next section is your proposed analysis. This section really, what I'm going to tell you for proposed analysis is really what I just told you, materials and methods. So I would just, as long as inside your materials and methods you talk about statistical analysis, data, the data tables you're going to collect, the data you're going to look at, then you can keep it inside the materials and methods. State the type of data you're going to collect, state the types of tables and graphs you're going to create. That's all within the materials and methods, right? State the types of statistical analyses you're going to perform on the data. State what you are expecting to see from the data, which will support your hypothesis, support or reject your hypothesis. The next section is going to be materials and costs. So you need to create a spreadsheet in Excel and create an invoice of all the materials you need, the cost for each item and how much of each item you need, as well as the total cost for each item, and then you need to finally tally the final cost. Make sure that cost is $500. All right, if you're having problems finding your supplies, commonly used science supply companies: Carolina Biological Supply, Ward's, Connecticut Valley for Live Organisms, Fisher, and Amazon.com. Bacteria Carolina also is very cheap for organisms, okay? The next section would be your reference section. This is a page containing all of your endnotes. The articles that make up the section come from your background literature search. Please use the Council for Science Editors format. I want you to using the sequence citation system. It's on our webpage. How to do CSE documentation on our webpage. Next is the abstract. Now it's funny, the abstract I told you at the beginning is the second right next to the title. The abstract is the last thing you write. Why? As I said, it's a summary of your entire proposal. So although the abstract comes after the title, it's the section that's written last. It's a summary of your entire proposal. It should contain a summary of the introduction, objective, hypothesis, and a summary of the materials and methods and proposed analysis. It should be no more than 2 paragraphs. Okay? Lastly is the appendixes. They are created to place any items that are too big to fit in the context of the paper, such as large tables, and figures. That would be really like if you have a table that requires 10 pages, then I would tell you to put an appendix. But most of you guys will not need to create an appendix. Figures, right? I want them inside the body. Okay, survey questionnaires you could possibly put in an appendix, or safety or ethical documentation such as IRB approval and stuff, that would be in an appendix. So they are found at the end of the proposal or final paper. In the methods section, be sure to refer to the reader to the documents located in the appendix if necessary. So I'm going to talk a little bit about scientific writing. Good scientific writing is concise and accurate and uses as few words as necessary. Use abbreviations only after the terms have been clearly stated first, right? So if you said Council for Science Editors and you put in parentheses CSE, then you could say CSE later on, okay? You need to use skills that you've already acquired. You have to remember paragraph organization, construction of topic sentences, and then support of those topic sentences with facts, right, that you got from your background literature, and then making sure that you cite where you got those sources from, and then proper spelling and grammar. The voice of research writing is a topic of dispute among scientists. This is new to me, but this is what I read in this book. All right, science can be written in active voice or passive voice. In an active voice, the subject performs the action. I, we, right? In a passive voice, the emphasis is put on what was done. Who performed the action is left out. 35 milliliters was placed into a 50 milliliter Falcon Blue Top tube. Okay? And just to make a point, the use of the second person is never accepted in scientific writing. I want you guys doing it in the passive voice. All the years of experience doing research and in medicine, you always want to write in the passive voice. Do not say we or I. This is done. 25 milliliter test tube was placed in a centrifuge and spun down at 10,000 RPMs for 15 minutes. The supernatant was decanted and the pellet was resuspended with 15 microliters of Helix. Let's say I want you all in the passive voice. Here are some examples of active versus passive of the pronouns in STEM research. So I will remove the ball bearing. That's active voice, right? I removed the ball bearing. That's active voice, but it's the past tense. I will remove it is in the future. The ball bearing will be removed. That's the passive, no pronoun, future tense. That okay? For your grant proposal, you're going to be saying the ball bearing will be removed. When you finished your project and you're writing your materials and methods, then the ball bearing was removed. Past tense, okay? Never the second person of directive. You should remove the ball bearing. No good. Or remove the ball bearing. No good. Okay? So we want to do the passive voice. When we write science papers, we want to do the passive voice. No I, we, or any of that stuff. This concludes this lecture on how to write a grant proposal. If you have any questions, feel free to contact me. Thank you very much for listening. Take care.